6 of the AP Biology exam, which covers gene expression and regulation. So here's some background. There are 4 nucleotides that make up DNA: adenine, thymine, guanine, and cytosine. In RNA, however, there is a 5th nucleotide called uracil, which replaces thymine and pairs with adenine instead. These 5 nucleotides have base pairing rules. In DNA, adenine always pairs with thymine, and cytosine always pairs with guanine. In RNA, adenine always pairs with uracil, and cytosine always pairs with guanine. These 5 nucleotides are categorized into purines and pyrimidines. As you can see here, purines have a 2-ring structure, and a pyrimidine has a single ring. And adenine and guanine are the purines, and the cytosine, thymine, and uracil are all pyrimidines. Looking at the width of the DNA double helix, there's only enough width or space for a purine to pair with the pyrimidine. If a purine pairs with the purine, then that's too big and that doesn't fit within the double helix. And if a pyrimidine pairs with the pyrimidine, that is too short and it falls short of the required length within the required width within the double helix. Regarding eukaryotes and prokaryotes, it's important to remember that they both use DNA and RNA to store genetic information, not just DNA. And looking at the chromosomal structure, prokaryotes have circular chromosomes located in the nucleoid. The nucleoid is this dense region where most of the bacteria's genetic material is located. And remember that prokaryotes do not have membrane-bound organelles, so this is not membrane-bound. Looking at eukaryotes, we can see that they have linear chromosomes located in the nucleus. So the eukaryotes have membrane-bound organelles, so they have a membrane-enclosed nucleus. And the nucleus is this where I'm circling right now, with the nucleolus being the dense center within it. So what is DNA replication? DNA replication is the process in which genetic information within DNA is passed on to the next generation. Note the difference between where replication takes place in prokaryotes versus eukaryotes. So as we just discussed, prokaryotes have a nucleoid, which is not membrane-bound, and eukaryotes have a nucleus, which is membrane-bound. So I'm going to be talking about a brief overview of these enzymes, which are most commonly found in DNA replication. There are more, but those are not as important. So helicase. Helicase basically breaks the hydrogen bonds between these nucleotides and unwinds the double helix. DNA polymerase catalyzes the reaction for the synthesis of the daughter strand of DNA and basically allows DNA replication to be possible. Primase lays down an RNA primer. So it's important to remember or know that DNA polymerase cannot start out of nowhere. It needs some starting point from which it can continue the synthesis of DNA. So that starting point is provided by the RNA primer, which is made by a primase. Next, topoisomerase. So when helicase is unwinding this double helix, there's an incredible amount of tension created in the upstream DNA. So within that DNA sequence, within that DNA, there's a topoisomerase can help relieve some of the tension and not have the DNA become supercoiled. So ligase. We'll get to that in a few slides. But ligase is very important for connecting multiple strands of DNA. And finally, SSBPs, which stand for single-strand binding proteins. As their name suggests, they're found on one strand, here and here. They're found in both strands. And they prevent DNA from pairing back with each other and keep the 2 strands of DNA separate. So DNA replication. There were 3 original models proposed for how DNA replication actually works: the conservative model, the semi-conservative model, and the dispersive model. So the semi-conservative model is the one that actually takes place, and the semi-conservative model states that the 2 original strands of DNA separate during replication, and each strand serves as a template for a new DNA strand, which means that each newly synthesized double helix is a combination of one old and one new DNA strand. So as you can see here, the red strand is the newly synthesized one, and the blue is the original one. In the semi-conservative model, as we just said, the red one new pairs with one old, and in the conservative one, the new DNA strands pair together and the old one paired together. And the dispersive one, the new and old just all mixed together. So here we're going to talk about the leading and the lagging strands. It's important to first know that there's a replication bubble, which looks like a bubble, that's why it's called the replication bubble, with an origin of replication where DNA replication starts. Within this bubble, there are two replication forks here and here. They basically indicate where helicase is unwinding the double helix and where the overall direction of replication is. So it's also important to remember that DNA polymerase works in a 5' to 3' direction on the daughter strand, and a 3' to 5' direction on the parent strand. So then, if you let's start looking by looking at the leading strand. So the leading strand, as we just said, works in DNA polymerase works in a 5' to 3' direction, and that's how the leading strand is synthesized, in a 5' to 3' direction, or a 3' to 5' direction on the parent strand. And let's look at the lagging strand now. So if you, as we just said, DNA polymerase works in a 3' to 5' direction on the parent strand. So that would mean that DNA polymerase has to work in this direction, as my arrow indicates. So if that's true, then how does DNA polymerase still work in the overall direction of replication? Well, the answer lies within Okazaki fragments. So these short fragments of DNA are called Okazaki fragments, and they basically allow DNA polymerase to work in the 5' to 3' direction on the daughter strand, but still move towards the overall direction of replication at here, or here if you're looking at this side. So within this, you can see that these red things at the start of at the 5' direction of each DNA strand or fragment, is an RNA primer, and that's laid by primase. So let's just start with how the lagging strand is synthesized. So if you start at the origin of replication, the lagging strand first starts. An RNA primer is laid down by primase, and the DNA polymerase continues replication from the 5' direction, as indicated here, to the 3' direction here on the daughter strand, which are these light blue strands. And then if you continue to the left, it goes 1, 2, 3, and etc. That's the direction of replication. This is the first one synthesized, the second one, third, etc. in this direction. So that's how DNA polymerase accomplishes that task of moving in the 5' to 3' direction on the daughter strand and still moving towards the overall direction of replication. So these Okazaki fragments, since they're fragments obviously and not a single DNA strand, they're connected by a ligase, as we talked about earlier, which connects strands of DNA. So looking at the central dogma, we just covered DNA replication. However, it's important to see DNA into RNA, and that's exactly what transcription does. The process of converting DNA into RNA is called transcription. So within DNA, you know that there are two strands of DNA. However, only one strand of DNA is always used for transcription. That strand of DNA is called the template strand, and it's always used. The other strand of DNA, called the sense strand, is not used. In fact, another name for the template strand is called the antisense strand. RNA polymerase, as DNA polymerase does, reads the template strand in the 3' to 5' direction. Thus, it synthesizes the RNA molecule in a 5' to 3' direction. Also important to note, after RNA or after transcription, post-transcriptional modification takes place, and that basically is in the form of a 5' GTP cap, which is just a string of guanine nucleotides, and at the 3' cap, polya tail or adenine nucleotides added to the 3' end. This basically helps the RNA from being degraded by the enzymes in the cell, and also helps with the ribosomal binding to initiate translation, which we'll get to in a little bit. Finally, alternative splicing. So alternative splicing. First, think about how many proteins a normal cell can produce. That's way more than the number of genes there are. So there are roughly 20,000 genes, and there are way more than 20,000 proteins. So the way that's carried out is actually through alternative splicing. Alternative splicing allows for many different combinations of protein products to be produced from one gene. So within genes, there are things called exons and introns. Exons are kind of considered the useful DNA, and the introns are kind of considered the garbage DNA. the introns are not used or not in the final RNA product. So as we talked about with the RNA, once it's transcribed, the RNA undergoes some processing like the post-transcriptional modification we just talked about. However, before that, there are things that splice out the introns or the junk DNA from the RNA that has just been transcribed from the template string. So after there are only exons left, it depends. Those enzymes that splice up the introns, it depends whether they regard which exons as introns, and if they regard different exons as introns, the final RNA transcript can be different than, say, another version transcribed by the same gene. And thus, many different combinations of protein products can be produced from one gene. Okay, so let's talk about RNA to protein, which is the final step of the central dogma. So to start off with translation, let's talk about ribosomes. And ribosomes are made up of a complex of proteins and rRNA, or ribosomal RNA. There are three major steps: initiation, elongation, and termination. Initiation entails the rRNA in the ribosomes interacting with the mRNA at the start codon. As you can see here, the start codon is AUG, and the complementary sequence, this is the tRNA molecule, and it has an anticodon, which is complementary to the start codon, and that anticodon is UAC. So the tRNA interacts with the mRNA and the small ribosomal subunit. Next, with elongation, tRNA brings the amino acid as coded by the complementary mRNA codon. So you can see here that the next codon, which is a triplet of nucleotides, UUC, pairs with the anticodon AAG. So with the AAG, the tRNA that has AAG, that brings phenylalanine, another amino acid. Finally, termination. Termination is basically when the ribosome reaches the stop codon in the mRNA, and basically the mRNA is degraded, the ribosomal subunits dissociate, and the polypeptide chain is released. So those are essentially the three steps of translation. Next, let's get to regulation of gene expression. So sequences of DNA have control over increasing or decreasing the expression of certain genes. They're called regulatory sequences. Other ways to regulate the expression of certain genes include epigenetic changes or changes not directly changing the base pair sequence. So in prokaryotes, they've evolved a strategy using operons. However, remember that operons are only available in prokaryotes and not eukaryotes. So again, there are two types of operons: inducible and repressible operons. So in inducible operons, they allow the expression of the gene by default and only turn off when the protein it creates is no longer needed. So the classic example of an inducible operon is the lac operon. The lac operon codes for enzymes that allow bacteria to use lactose for energy. But as we just discussed in the description of an inducible operon, bacteria do not want to use lactose for energy unless glucose is not available, and that's because glucose is their preferential source of energy. So if glucose is not available, only then will they start transcribing these genes. So that's thus fitting the definition of the inducible operon, where it's always turned off and only turned on when needed. So the lac operon is regulated by the lac repressor protein and the cap protein. The repressor protein binds to the operator region of DNA, and the promoter region is actually where RNA polymerase starts. It attaches to and starts transcribing. So as we can see in the diagram, in the absence of cyclic AMP, cap does not bind to the promoter, and transcription occurs at a low rate. In the presence of cyclic AMP, cap binds to the promoter, increases the RNA polymerase's affinity to the promoter, and it promotes transcription. So next, repressible operons. Repressible operons are normally not active and only turned on when needed. So as you can see as in here, the TRP operon, tryptophan acts as it's called a co-repressor, which binds the repressor and helps it to limit transcription. So as we can see in the diagram, in the absence of tryptophan, the repressor dissociates from the operator and the RNA synthesis proceeds. The repressor needs the co-repressor to be active and bind to the operator, thus limiting transcription. When tryptophan is present, the TRP repressor binds to the operator and RNA synthesis is blocked, as you can see here. Gene expression. To sum up, gene expression affects cell products and protein function, and regulation of gene expression at any level, be it epigenetic changes or post-transcriptional modifications, or even modifications at the protein level, like adding carbohydrates or lipids or anything else, will result in differential gene expression and thus a different phenotype. Okay, so let's talk about mutations in the genetic code. So there are three main classes of mutations: insertions, deletions, and point mutations. Insertions and deletions are frameshift mutations, and the entire structure of the resulting protein may be drastically altered because of these insertions and deletions. So as we talked about, when RNA is being transcribed, it has these things called codons, which are in fact triplets. So if within the DNA sequence, if one nucleotide is added or removed, it'll disrupt this how the RNA is transcribed, and since the RNA sequence is changed, it's altered by adding one or subtracting one. The sequence of codons basically shifts, and the entire sequence after the mutation changes, thus potentially making the end protein drastically different than the original protein. The next class of point mutations. So point mutations refer to a single base pair substitution, and they can be silent, missense, or nonsense. As we saw in the RNA codons, so there's within the last nucleotide of the codon, there's a little bit of leeway as to what it can be, as multiple codons can code for the same amino acid. That's called the wobble effect. So with the wobble effect, multiple codons can code for the same amino acid, yet even though they have a different last nucleotide as part of the triplet. So if the last nucleotide is changed, say from a U to a C, for example, it still may code for the same amino acid as the previous one did. In missense mutations, the mutation makes it so that the mRNA codon codes for a different amino acid, and in nonsense mutations, the mRNA codon codes for a stop codon, essentially ending the protein there and stopping translation at the stop codon. Okay, so let's talk about a little bit about DNA and biotechnology. So PCR, the main things are PCR, gel electrophoresis, bacterial transformation, and DNA sequencing. PCR is when you take a DNA sample and amplify it using polymerase chain reaction, which entails a bunch of enzymes, and it's not as important to discuss the details. With gel electrophoresis, that's when DNA molecules are separated by size and charged in the gel by running a current through it. Bacterial transformation is when DNA from other species are introduced to bacterial cells to make them express a new protein product. So for example, with insulin, bacteria were introduced to the gene that makes insulin, and that's how we actually make insulin commercially. With DNA sequencing, that's pretty self-explanatory from the name, but DNA sequencing analyzes the order of the nucleotides within a segment of DNA. So finally, let's just get to some practice questions. So with the explain how the process by which DNA makes proteins. These questions actually appeared in prior AP exams. So just pause if you would like to answer it yourself, and then unpause if you'd like to check your answer. So how DNA makes protein. So the first stage of the process is transcription, where DNA makes mRNA that carries the message directly to the ribosome in the cytoplasm. So transcription basically entails RNA polymerase and the DNA template strand. Remember that only the template strand is transcribed, not the opposing strand or the sense strand. So after transcription, DNA is made into RNA, and RNA processing like alternative splicing and post-transcriptional modification takes place. So with the post-transcriptional modification, that basically is adding a 5' guanine cap and a 3' poly A tail. So with the 3' poly A tail added, so that basically finishes post-transcriptional modification. With the alternative splicing or splicing out in general, the introns, as we talked about, or the junk DNA is removed, and only the exons are kept. With alternative splicing, it depends by those enzymes that do the splicing, which exons are treated as introns and which ones are not. Finally, let's just get to RNA to proteins, which is the last step of how DNA makes proteins. A translation is the process by which the codons of an mRNA sequence are changed into an amino acid sequence. Amino acids present in the cytoplasm are carried by tRNA molecules to the codons of the mRNA strand at the ribosome according to the base pairing rules A with U and C with G. Remember that A and G are the purines and U and C are the pyrimidines. One end of the tRNA molecule bears a specific amino acid, and the other end bears a nucleotide triplet called the anticodon, as we discussed. Some tRNA molecules have anticodons that can recognize two or more different codons. This is because the base pairing rules for the third base of a codon are not as strict, and that's the wobble effect, as we just talked about. Okay, next, the translation of the mRNA strand is complete when the ribosome reaches one of the termination or stop codons. After it reaches the stop codon, the mRNA is degraded, the tRNA is reused, and the polypeptide is freed from the ribosome, and the ribosome dissociates. So essentially, that's more or less the answer for that. With this short free response question, in what way does pre-RNA differ from mRNA? Explain why the two are different. As we just talked about, there's a post-transcriptional modification, and it's mainly just a post-transcriptional modification and the alternative splicing. So with the post-transcriptional modification, again, it's the 5' guanine nucleotide cap and the 3' poly A tail. Again, with splicing, the introns are removed, and the exons depending on which one exons are treated as introns, those exons that are treated as introns are removed, and the rest are remain. So those are the two ways in which mRNA differ from pre-RNA. Thank you.